Hello, my name is Michael Antonov. I'm an entrepreneur and biotechnology investor of Formic Ventures, and also recently founder of Formic Labs. And due to my long history in software, which included Oculus, I got interested in computational modeling of biology, which is really the question of how far are we from being able to simulate life. And this ultimately led me to learn about the work of Markus Covert, who's really built an exciting and passionate career in the field. And the way I learned about him first was from his leading work on the first full cell model of a bacteria quite a few years ago, so which led me to connect with him. And today I'm excited to be joined by Mr. Markus Covert, who is in fact now a chair of bioengineering at Stanford. Markus, thank you so much for joining me.
Of course, it's a pleasure to be here and pleasure to just be able to chat.
All right. So the first question comes to my mind is, what are the full cell models and what can we learn from them? Why would we want to build them?
Yeah, it's a perfect and a critically important question. This century, I think, is going to be the century of biology writ large, not just in terms of human health, but I think there's a huge motivation there. Maybe you've seen these new governmental initiatives, ARPA-H and others, where they're really thinking a lot about how to make health a primary advances in cancer and other diseases over the next many decades, but also now thinking more about sustainability, biodiversity, the planet, planetary health, like all of those things are coming right at us, in rapid motion. But unlike many of the other engineering disciplines, we don't have the same modeling and predictive and design capacity, computational capacity in these fields that we have had in previous spaces. And my favorite analogy is weather prediction. So I had dinner with some friends a little while ago, and we were trying to argue what's the most important thing that the human has happened in human civilization. What was our biggest contribution? I think one of those was our ability to predict the weather, like a hurricane, 10 days in advance, with enough time to save lives and save property. When you think about what that entailed, people had to work out mathematics of a highly nonlinear, like very chaotic system with random events that happen. They had to invent chaos theory. They had to invent satellites. They had to make measurements all around the world. They had to make measurements at large scale and small scale. They had to put these things together. It's a massive human achievement, but it's been transformative for us. And if I were to say, what's the big next achievement? So that happened in the 1900s, last century. The big next achievement for this century is something along the lines of, if we could predict a hurricane in 10 days in advance, can we predict a tumor 10 years in advance? That's the great challenge that I think we should all consider. And when we do that, just the same way with satellite technology and all these other, it'll have lots of follow-on effects, lots of follow-on impact. And yeah, so that would be my take on what it is, why it's important. I can describe the nitty-gritty details about what we've actually done with the cell if you'd like for me to, but that's the field, if you just think about it in a nutshell, what we need to be able to do.
I mean, one of my thoughts was if we can actually model a cell, then we could, let's say, compare it to maybe a disease cell or an aged cell. And if we have the model, we could try things on it, whether it's novel molecules, whether it's some kind of genetic, epigenetic modification, and see if we can improve the state.
Yeah.
Is that kind of something which can help us with drug discovery and understanding?
That's a beautiful idea and insight. So the reason why we're working on making models of single cells, right, where we take all of the genes and all the content and we try to see if we can model how genes are expressed, how different things in the cell happen, how it grows out and divides. if we can do that for one cell, and ideally for groups of cells, and then finally for tissue, for a tumor, something like that, then you're exactly right. We can take all of that information. We can take a simulation, let's say, of a tumor cell, and then we can say, well, what happens if you try this drug? What happens if you try this combination of drugs? What happens if you change your environment? All of those things are things that are impossible for us to do right now, but that will become more and more possible as we expand out this technology.
Mm-hmm. So I wonder, so you've built the full cell models and you've done a whole lot of research in Zen. I understand you've built the whole colonies of cells. I believe the vivarium is a project you're on. What are a few things that you've learned from building this?
Yeah, I would say there are a few things that have come to the forefront. Like, obviously, we've learned a lot of things about the technology, but I'll focus on the biological insights. One of the most interesting things that we've learned is the role of stochasticity, which would be noise, just things that happen in a cell. The cell has to deal with the fact that a lot of random events are occurring. You just have a small number of molecules, and that means that a certain event happening can have a big impact on the cell. And the cell has to be ready to respond to that, right? It has to be able to maintain what we would call homeostasis, right? It has to be able to do its normal program, even in the face of all of this noise. And that's something that I think the world is starting to recognize more, the biology world, how cells manage to do what they want, even in the context of these random events. But I think it's also become clear that cells even sometimes exploit and create that noise for their own purposes, right? And so one thing that we have found is that cells generating their own noise, their own variation in behavior, is one of the ways, for example, that bacteria can evade antibiotics, right? Or that a tumor can evade a treatment. So those kinds of things are very interesting. I could go on and on. We could probably talk about the insights for 10 hours, but that's one of my favorites right now.
That is fascinating work. So I clearly came into this with this biological interest, and as a software engineer, one might think, why can't we just simulate everything from atoms? So, like in computer code, we have these assembly instructions, we can all learn from them, and this whole incredible world of software comes from it. And one would think, well, okay, biology comes from a world of chemistry and reactions and atoms. So if we just had a really good simulator of atoms, then we could model everything, and perhaps we don't need this heavy lab equipment in the future. Now I've learned that things are not quite so simple, but I guess my question to you is, there's clearly these different levels of detail you can study biology at. And the lowest one is atoms, and there is a whole field of molecular dynamics simulation where you computationally do that, but it doesn't always work for many things. So I'm curious, how do you think about like molecular dynamics simulation? What can it do? What can it not do? And when do you need to be in a higher level for like really understanding biology?
Yeah, this is a great question. So when I was an undergraduate, actually, I did my research was all in molecular dynamics. And so we would do, just like you say, atom simulations. We would have, let's say we have a couple of atoms here, and I kind of think of these as like tennis balls bouncing against each other in a room. That's how Richard Feynman, the physicist, described, his visual paradigm, and it's definitely infected me. So you imagine these different balls bouncing around, they're moving with velocity, and they have a certain attraction or repulsion with each other, right? And back when I was doing it, we could shut down all the computers basically on campus, just running these, these nanosecond or femtosecond simulations of these tennis balls bouncing against each other, basically. But the goal of it was to try to determine, in our case, osmotic parameters, right? Like to be able to determine how cells want to move from one place to another. And they can be very powerful in that area. The complications with, and especially when we start looking at how proteins fold or move around, molecular dynamics has a lot to say in some of these areas, which is great. I think the complication is, one is just the sheer compute time, right? And another is just the specificity. So, when you are doing an all atom simulation, there's no way that you can make a direct comparison to a direct experiment, just exactly because of all that noise and variability that I was talking about, right? There are too many degrees of freedom, too many things can happen. And so it can be extremely important for helping us to determine parameters or properties, right? That's where I see molecular dynamics. Maybe if I were to go back to my weather analogy, right? So for the weather analogy, imagine now that you say, well, why do you need satellites, right? Why can't I just, let's say, have a very high-speed camera and throw some dirt in the air and watch how the wind moves it, and watch every grain and particle and see where it goes, and map that, and then use that, extrapolate from there to say how all the weather in the Earth is going, right? So what I would say is, well, we should be looking at how dust moves around in the air because it will teach us principles of fluid mechanics. It will help us to understand properties of how things move in this kind of fluid air by weight. It tells us a lot. We can learn a lot, but it's not the right scale to consider modeling the entire Earth's weather patterns, right? For that, you have to take a step back. You take what you've learned from this modeling scale, and you then apply it, as you move up scales, you take those properties as opposed to the motion of every particle of dirt, right? The motion of every lamina of air or just the turbulent flow of the molecules in that way.
So we learned some of the parameters, but I guess it is around 1 trillion atoms you'd have in a cell, or somewhere in that order of magnitude, right? So it's just computationally expensive.
That's
and
right.
The Moore's law hasn't quite gotten us there yet.
Yeah. And probably, going back to my weather analogy, probably not even the most useful framework for us to consider it. That, you know what I mean? It's useful, but it shouldn't be the entire framework. Just the same way we wouldn't want to model all of the specks of dust moving around in the air around the Earth.
So I guess at this level, we can learn maybe how molecules and proteins bind, how some of them might get arranged. And then what is the next level up? Like, how do you use that information in a higher level context?
Sure. So here's a great example, right? So we have had a massive transformative revolution in AI, right? AlphaFold is now able to predict the protein structures. For example, in E. coli, the simple bacterium, that we do most of our current cutting edge modeling in, all of the proteins, at least a structure, has been predicted by AlphaFold, right? And many of the structures have been validated against actual known experimental structures. That's great, but one thing that AlphaFold cannot do is tell us what happens if there's a mutation in the gene sequence for that protein. And that's super important because when we treat bacteria with antibiotics, for example, the way that they get around it permanently is by making mutations in their own DNA, typically. And so we would really like to know what happens if there's a mutation in a protein. How does that change how it can bind to the antibiotic, let's say, right? And so at this is where my molecular dynamics could be amazing, because you can start with this protein structure from AlphaFold. You can add the mutation. You can let molecular dynamics kind of figure out, now, here's how the protein changes its structure. Maybe this thumb goes in the middle now, right? And now, let's say this is the antibiotic, whereas before I could hold the antibiotic here. Now, when I move my thumb, because of the mutation, the antibiotic no longer fits in there. And so I'm free, like I'm resistant to the antibiotic, right? That's the kind of thing you would find for molecular dynamics. AlphaFold would give you this. Molecular dynamics would give you this. And then what I would do, I would take this as a numerical value, a binding constant between
Okay.
an antibiotic and a protein. And I would then say, okay, what does this do for the whole cell? How does that bring the whole cell together? And what does that mean? Does that mean now that it's resistant, or does this antibiotic hit other things, right? Or does it have another impact that we didn't know about? That's what the cell model will tell you. And then what the colony model will tell you is the fact that different cells have different sets of genes that are turned on and off. And so this mutation might have only happened in one cell in the colony. And so even though this one cell can escape, the rest all die.
So maybe that cell can divide and form a new colony.
Exactly.
Or not.
So now you see how like all these scales, they're all important and they all fit together. In my lab, we've chosen the scale that I think most close, like gives us the most possible utility, but they're all important.
Mm-hmm. So I'm interested in the role of AI on all of this. It's clearly AlphaFold was a huge achievement. Now there's a whole different dimension with GPT and language models. And another maybe less heard advance that I've learned about a few years ago was ability to learn energy fields, which speeds up aspects of molecular dynamics we talked about more. So you can maybe get 1,000 or 1,000,000 times improvement in some area, which gives me hope of maybe we can combine all of this. But at the same time, I don't know, can AI, it's hard to get a sense of the limits of what an AI can do. Like, could you just have one deep neural net and just tell it lots of data about the cell and forget about all this physics and all this internal complexity we've been spending time on and just say, you know what, this will just do it for us? Or are you going to miss some fundamental unexpected change? And like, how could these things be combined? What is the limitations in each?
So one thing that I think, right, it's kind of like that famous quote, it's very hard to make predictions, especially about the future. So
True.
in this particular case, you know, I think there were people who thought that the protein structure problem would never be solved, right? And they certainly would have said never by AI, right? Like that was a shock. And so it would be very foolish of me to say a priori, like, no, AI can't do anything for us, right? I do think instead of maybe saying nothing could happen, I could talk a little bit about the challenges that exist that could be helpful for AI to consider, you know, as people try to move into that space. One challenge that I think is significant is that in a lot of fields, we don't actually care what's on the inside. We just care what's happening on the outside. So like the famous example of, you know, being able to recognize a cat, a picture of a cat, right? I don't care how they recognized it was a cat. I just want to know if it's a cat. And so you can imagine for AI, let's say we're looking at some scans of different patients, you know, and we see a tumor, or let's say it's very small. And so the AI says, oh, this is a tumor. And I was like, oh yeah, I didn't see that. But now that you say, I think it is a tumor, right? And in fact, people from my lab, alums from my lab, like David Van Balen at Caltech, are doing great work in this exact space. So the trick is, in this case, I'm also curious why. Like, I want to know what's on the inside. What happened that made that cell become a tumor, right? What was the internal mechanism? And so, you know, my lab has definitely worked in AI. We love it. Like, I'm a fan. At the same time, in this particular case, I think the mechanism is really, really important. And so we'll want to come up with methods. And one person that does this is Jason Yang, for example. He has these kind of mechanism inclusive neural nets that he develops that are really interesting. I think that could be a really exciting space. But my hope is that we'll come up with ways to make the mechanistic modeling, which includes the reasons, right, which includes the underlying mysteries, and we'll make those interact with black box, just neural net, deep learning. I'm very excited about some reinforcement learning possibilities, and of course the generative, like you said, ChatGPT, et cetera. Very exciting to see what's potentially out there. And by way of connection, I am, like so many of us, right, super inspired by Turing's original idea of an imitation game, right? Like, what if you or I, even though we're talking to each other, we're actually just a robot, which is completely computer generated, right? Like, what if everything you've just heard from me is complete video deep fake and just ChatGPT talking through me, right? I'm sure that's going to be a future that we're all looking at, right? But ChatGPT is kind of the closest we've gotten to meeting Turing's idea of an imitation game, like where really it would be hard for a human to tell that is not a human. Obviously, it's not super sophisticated, but it's still miraculous as far as it goes, right? I think it's quite amazing. Interestingly, to me, like, our models have also, and our simulations have started to hit a point of an imitation game. And what that means is, like, in our recent colony modeling that we have, our recent images, I've spent decades of my life looking at microscope data, looking at fluorescence proteins, looking at individual cells. And yet, when we saw our first whole colony simulations that we're just about to submit for publication now, I would have thought those were real, right? I would have thought that was a real microscope image with real fluorescence. It didn't last too long. After some seconds, I was like, oh yeah, this is a simulation. But to me, that's been a real turning point in I've always been very optimistic and enthusiastic about our methods, but for me to just see that and realize, wow, we're simulating life in such a way right now that I myself am falling for it. That was very exciting.
That is very exciting. You mentioned microscopes, and clearly being in biotechnology, I've learned that you really do need lots of real wet lab experimentation in all areas. And of course, when you work with humans, you ultimately need clinical data and many of these things. What do you see the role of experimentation or high throughput experimentation in this modeling world? Like, do we need both? How long we will need both, and at which level?
Yeah. Okay, great. This gives me a chance to maybe put some things together, right? So my vision is that one day we will be doing far more automated experimentation, modeling, and learning. Okay, so one vision that I have for discovery in the future, thinking about like something like ChatGPT, would be something like this. We need to do this experimentation, but it's not very efficient the way we currently do it, right? Lots of labs do it independently. The data isn't shared adequately, right? We need things, but there are a lot of things that we've looked at in great detail, but they're big gaping holes where nobody's looked at all. All of this kind of stuff could be directed and systematized by using modeling, just like it is done in other practices, right? Like chemical engineering, they have a whole field of trying to figure out the most information with the fewest experiments, right? These fractional designs, right? It's incredible. So I would say my favorite vision would be something like this. You generate a high throughput data set, let's say. This will be the first step of the loop. That data set for most humans is going to be impossible, even highly trained humans, is going to be impossible to interpret because there's just too much information and there's too much emergence going on in there. It's a complicated system. So what you do is you use a model to do the same experiment, but you do the experiment as a simulation. Now you compare the results of each, and many of the times there's going to be overlap. They're going to agree, but sometimes they won't agree. So now this is where something like learning and assisted learning could be amazing, because what a learning and data analysis can do is find those discrepancies and then start to say, okay, what is the most important experiment to do next to interpret what just happened, right? And maybe just like prioritize, okay, we saw these discrepancies. If you make these changes in the model, it will explain some of these discrepancies, right? And so you can imagine a loop in which you're generating data, possibly even in a totally automated way, no human, right? Just automated, like these cloud labs. Experiment, model-based comparison and interpretation, AI-based generation of new hypotheses and new path forward, right? Which then guides the experiment, which guides more simulations to help analyze and figure and interpret, which then leads to more generation in this kind of human out of the loop discovery paradigm. Now, some people would say, well, what about the massive creative impulse, right? Would we ever discover, transposable elements like Barbara McClintock did, just looking at single kernels of corn that looked funny in a whole corn cob, right? Like massively transformative insight, but something you'd never find. Yeah. So my hope is that if we generate these kind of human out of the loop discovery machines, we'll be learning a lot. And then we free up the most creative humans to be able to do that incredible out of nowhere, super creative work that really is something right now, and I believe for a long time in the future will be fundamentally a human enterprise.
Yeah, I mean, this is certainly an area where I think a number of companies and visionaries are going. Like I know in Silicon Medicine, and I'm good friends with Alex, the CEO, they've built a robotic lab and they're trying to do it in a loop. And there's a number of other companies which have built these automated iteration pipelines. I'd say it's not yet at scale or broadly accessible. So there's a whole lot we can do. I'm curious, so if you have this automated screening, can you actually set up some process which would let you fully understand and comprehend the underlying pathways in the cell? And pathways, this relates to gene expression, how much of which molecules generated. I know, just kind of introducing it a little bit. And I know that's what you've worked on a lot. But I know that there's a shortcoming. We don't know the genes. We don't know the kinetics of how well each protein works. Could a system we built, which through some set of automated process lets you fully understand, let's say, a human immune cell or something like that?
I completely believe that's possible, right? So I think I told you before, like if we can predict a hurricane 10 days in advance, can we predict a tumor 10 years in advance? I was at a giving a talk not too long ago, and I said that same thing, right? And somebody yelled from the back of the audience, "No, we can't." And I thought that was great, because I thought, he's right today, but he's going to be wrong tomorrow. Like there's no reason, given everything we've been accomplishing in this last years, that we shouldn't be able to build whole cell models of very complicated cells. It's going to require some concerted effort. Like if you look at the weather analogy, which I think is our closest analogy, right? The weather, I think in the first volume of Science, so the first year that the journal Science existed, one of the articles was a call, and the specific words were this: that call for the mathematician and the physicist to consecrate themselves to this work, right? So they've been working on this for over 100 years, right? It was a massive effort. I don't think it will take us 100 years, but you just get this sense they worked to develop the data, to develop, and the entire world, even countries that are at war, still share weather information with each other. That's like our best treaties, I think, in the world are around weather sharing, because everybody knows that we all need it, right? Like we all need it. It doesn't work. You can't just simulate weather patterns in Palo Alto and somehow from all of that figure out what to expect next. There's just no way to do it. And so we all have to share. And I think similarly, you know, right now my lab and a set of other amazing labs are working in this space, right? But we need something bigger. The first person to ever call for this, by the way, was Francis Crick, right? So in 1973, Francis Crick said, we need a worldwide effort to make a complete solution of a single cell. And that cell was E. coli, right? That's the one that we will do with the community together, right? Like we as a scientific community, we will do that. I would say in the next 5 or so years, we'll have something that takes 90+% of the genes into account and can predict many different environments, many perturbations. And then I think you're exactly right. We should go either to immune cells. That's a great place to start because we already have then a bacterium. So we could maybe, and we have a dynamic environment because of those colony simulations. So you could imagine starting out by just making the environment more living, like maybe it's the interior of a cell as opposed to an agar plate, right? So start thinking about how to make that environment more realistic. And all of our simulations these days are built on containers. So eventually you could just make that environment a container that was actually a cell, like an immune cell.
Mm-hmm.
But I also think there's great work that could be happening in brain cells, like neurons. I think would be amazing. As you know, I've been talking to different groups about this now, and I have a lot of optimism, including from those groups that if we had the will, right, and if we had people who were willing to kind of embrace a larger scale effort to go after this, we would do it, and it would be transformative. And
Yeah,
again, we doesn't mean me, it means like
the
everybody.
community.
Mm-hmm.
Well, since we're talking about predictions, there's like a couple of other interesting milestones which come to mind. So first, for me, I think it's very exciting is will and when we'll be able to solve full molecular self-assembly. And what I mean by this is protein complexes binding to, say, DNA and RNA, so fairly complex system self-assembling. Do you
Mm-hmm.
think and I know AI and AlphaFold comes in, physics come in, unknown research. If you were to make a guess, how far away do you think it's doable this decade, or might it be a bit longer?
Yeah, there have been some limited efforts in this area, mostly around protein folding and protein antibiotic interactions, right? It gets more and more complicated as you add multiple pieces, right? So for example, there was a deep learning based algorithm that attempted to predict kinetic parameters based on protein sequence. And it's pretty good actually in some ways, but it only works for monomers. So you can't do complexes yet. And of course, most proteins complex in one way or another. So I think some of the best and most exciting work is happening in that area. And of course, with people that we both know well, that are working in this space, it's an exciting frontier. And I think it will have this huge benefit of providing, kind of like we talked about before, providing parameters and insight that will then go into these cellular and multicellular models that really could be transformative. So I've even seen there was a company that worked on this in a way that I thought was really profound. They used network modeling to try to figure out what would be the best part of a network to target with a drug. Then they used the kind of modeling you're talking about to figure out how to design a drug to fit in the binding pocket or wherever in the protein. I don't remember all the details. And then they further went and found, using personalized medicine and single genome analysis, they were able to find the target population that would be best for this intervention. And that's the future, right? That company was ahead of its time, but that's the future of medicine, I believe.
Mhm. So now, how about the full cell human cell? When can we have several fully digitized human immune cells which respond to stimuli and predict reaction to drugs or bacteria or COVID? I don't know.
Yeah,
How far away are we?
question. It's a really good question. I think it kind of depends on who comes into the space and how our training works, right? So my lab is unusual because I mean, you know the kind of people that you know, several of the kind of people that join my lab, right? They tend to be extremely skilled in multiple areas. I always say this as being intellectually ambidextrous, right? So they're able to come in and write their own software, like that's pretty great, and also do their own models and run their own experiments. And so they have this like deep insight that right now is still pretty rare. There are some disciplines that teach this and some schools that teach this really well around the world. But we need more people who can kind of do this, or at least come together with like-minded or individuals with different skill sets and teach them how to collaborate. You know, right now, as you know, like my lab's kind of driving the cell level modeling of this things, although there are other awesome efforts going on in other spaces, including at different scales, right? But we need there to be, you know, 10 dozens at least of labs that are working in this space, like actively competing, scooping us, you know, doing all this stuff so that we're all driving, really accelerating the education, right? Because I can only educate people in my lab, people taking my class, right? Same thing for everybody else. We need to just grab a community, train them, inspire them, and set them loose. And then I think, you know, sky's the limit, right? The reason why AI is moving so fast right now is not only because of the compute, but also because it has the interest of some of the brightest people in the world.
Yes.
Right? That is a key thing to remember. And I think that that we also have the interest of some of the brightest people in the world, but not at the same scale as AI has right now.
So yes, I mean, what I'm excited for is actually companies and more people, not just academics, getting into it, so that we can have a growing ecosystem.
Yeah.
And hopefully more automated, shared, accessible tools like you described.
I agree. It has to be open, and it has to be. It's funny how sometimes fields grow up and they get really competitive and unfriendly. I'm a big believer in being competitive and friendly. So making stuff available,
Yes.
right? This is how the early molecular biology field got founded, by the way. They were all buddies, you know, but they were also fiercely competitive, and that's a really good ecosystem.
Mm-hmm.
Yeah.
So before we wrap up, are there maybe any other areas of biology or something which you are excited or just like thinking about as you know that are developing now?
Yeah. Well, I think it's connected, right? So as you mentioned, I've been the chair of the bioengineering department now for 4 or 5 weeks. And it's been interesting to see what people
Congratulations, by the
are.
way.
Thanks, thank you. It is an honor to be part of what I think is, you know, the greatest department in the world. I love our department. But one thing that's been interesting for me is to see at a higher level, like what people are looking at as the major challenges of our time and how they're coming to address them. One of the major challenges, or one of the major opportunities, is really kind of the same way that AI has been transformed in recent years. Synthetic biology has also been transformed, right? Truly transformed thanks to CRISPR and just the rapid innovation in the space of CRISPR. And of course, many of those people are in this Bay Area, right? We have some of the most important players in the space here in the Bay Area, including in my department. And So when you think about it, this is comparable to like the invention of transistors, right? Or some crazy, like where computers, personal computers, like what is this going to do for education now that we can change biology, right? Like just by altering genomes, right? What is this going to mean for agriculture, right? For good or for bad, right? What is this going to mean for sustainability? What is this going to mean for health? In every other field, they have computational tools to help you predict what's going to happen so that you don't blow up the factory, right? So that you don't make a massive, dangerous decision, right? Or a very costly decision. And so I'm a huge fan of what's going to happen with synthetic biology, but I urge the community that we have to also come up with predictive tools so that we don't create biological disasters, right? Like we have to be careful that we're not doing things that are safe and well reasoned, and where we've minimized the side effects and optimized the benefits and advantages. The time has never been more important for us to come up with methodologies like that. So I see huge opportunity, huge positive opportunity in synthetic biology. And I also see huge need and necessity for exactly the kind of tools that we've been discussing, not just for discovery, but now moving it, and this is maybe a good way to close our discussion, moving it to design, like moving it to design tools from discovery tools.
I mean, that's exactly feels right that these two areas complement each other. On one hand, you edit it, and on the other hand, you have computational simulation machinery to really understand what does it do. And you couple the two together to make sure everything goes well and you learn from it.
Yeah.
Well, it's a pleasure, Marcus. I feel like this is super exciting decade. So I look forward to learning about what your lab comes up with and how progress moves forward.
Yeah, thanks, Michael. And likewise, like I've been really inspired to see you kind of moving from this very successful experience with Oculus and other things to really kind of taking your passion and making something happen out of it in a field which is relatively new for you. So that's best of luck in that journey, and anything I can do to be helpful, let me know.
All right. Thank you, Marcus. Have a good one.
You too.